No, I don't see any other. No, I don't see any way to sort of. Okay. I hope people are able to join because I had been clicking join for the past whole 1 week, but seems that everybody wants to join on their own sweet time, and that makes life very difficult. Essentially, let us start with, because at least I should not work in favor of people who are not serious. At least those of you who are here should be given a better chance. So now, today's class, my students will take over at some point of time. So the point really is rather simple, though we have practicals which are online, and online practicals are a bit of a misnomer. It makes little sense. But all the time we are talking about the vector plasmid, and we are drawing a vector plasmid here, a circular plasmid, and then we are taking up insert here, which is the gene of interest, and sort of inserting it here, and so on. And then we are taking this whole plasmid and all this and putting it inside the cell. Now, really, how do we know this is happening? After all, you can't see the gene. You definitely also can't see the vector. How do you know it has got joined inside? How do you know this combination is taking place? How do you know it has gone inside the cell? How do you even see a single cell? You don't. Forget the vector and the gene. And then, of course, this vector and gene insert would possibly have a promoter here, and so this should be making some RNA. How do you know it is making RNA, and how do you know it is making protein? You can't see the individual protein peptide, polypeptide, and so on and so forth. So, can you hear me?
Yes.
Yes, sir.
Yes, sir.
Yes, sir.
I think there's something wrong here. Okay, let me minimize this and let me sort of we should keep this thing as on. Is it not showing up? Okay, so whether we are seeing the protein or and all these things have to be seen on the gel or by various techniques that we need to know. Now, what are those various techniques which allows us to visualize the gene? Allows us to visualize how the gene enters the vector. How do you visualize that the vector plus the insert, which is called a chimeric plasmid, enters the cell? How do these cells grow and produce the RNA and then produce the protein? Then how do you sort of course, see where the protein has gone inside the cell? Is it still inside, or is it outside, or is it in something called the periplasmic space, and so on and so forth? This is a problem. This is a real problem. Anyway, so it could be in the periplasmic space, which is in the there's a periplasmic space which is in between the inner membrane and outer membrane. Can you hear me?
Yes, sir.
Yes,
I
sir.
can't see this. Why can't I see this? Yeah, now it's better, I think. Let me see this. Yeah, so now I can see that the channel meeting is on. So now, what are the techniques which allows us to visualize the gene, the DNA, the vector insert, whether it has got inserted, whether it has gone inside a cell, whether the cell is producing the protein or not, and if it is producing, where is it going? If it is not producing, how do we know it is not producing? What could possibly have gone wrong? These are the stuff that practicals are made up of. And these are not easy questions. They may appear very simple, and over a period of time, these jobs have indeed become simpler with the emergence of kits and much better equipment, but still it is quite a lot of art and only a little bit of science. So I would suggest that you begin paying attention. My students here will demonstrate each and every step, but it will be like a cookbook. They will cut out all the intermediate waiting steps because obviously there is no time and makes no sense in a practical which is demonstrated online how this is to be done. So I suggest you listen to it, and at the end of the day you are going to write up a report. There'll be approximately 10 practicals. Each practical will be a report, a brief summary of the individual steps that have been taught to you and their descriptions. And that carries 50% marks. The 10 practicals carry 50% marks. We will have a mid-semester exam, which will be 20% marks, and an end semester exam, which will be 30. But the lab practicals, the lab notebook carries 50% marks. And they will be evaluated and you will upload each and every practical on Moodle and we will check it out. So this is your job, and I hope that both the 2018 batch and the present batch are both doing this course. I'm assuming that all of you are doing this. This practical should be submitted before the next week practical, that is before Monday tomorrow. I don't want you to wait, and I don't want any copying. This has to be in your own words. So if the language is the same, if the words are the same, then sorry, you get a 0. And there are 10 practicals. Let us say each of these has 5 marks, and you'll be given marks according to how well you have understood it and how well you have written it. We don't want you to write long paragraphs. A couple of pages, 2.5 pages, typed pages, broken down into individual paragraphs about individual subjects will be enough. How do you see the protein? How do you see the gene? How do you see the vector? How do you ensure that expression has taken place? How do you know when expression has not taken place? Where has the protein gone? 5-6 questions. You have, because I don't want to, the reason I don't want to tell you what those questions are and how to answer them is that I want you to divide up these practicals into those 5-6 component parts on your own, so that not everybody divides it in the same way. Uses his or her own language to divide the practical into 6-7 small paragraphs and descriptions thereof. 2.5 pages on your own to show that you have understood what is going on. Is that clear? Can you hear me? Is it clear?
Yes, sir.
Sir, can
So
we
we will
type
start.
it?
Sorry?
Sir, can we type our reports?
You have to type your reports because it is
Okay.
better. Otherwise, how do we read it? Okay. Please upload
Sure,
it instead of taking photographs. Please type your reports and send it. Right? 2 pages. Type 2
And
pages.
sir,
If you
we
can't,
have
then
to
you
upload
can
it
write
on
it down.
Moodle.
Yes, and upload it on Moodle every week. Last week we are giving you a pass. This week, no more passes. And we will put the video on and the camera on and actually let you see rather than do it. So I'm going to pass it on to 2 of my senior students, Dr. Manjul Tripathi and Dr. Arunangshu Das, who are going to take in a lot of time off to prepare these practicals. So please listen carefully. I don't want people running away the way you people do. So I will, Arunangshu and Manjul, why don't you take over? I can put off this.
And sir, one more question. Sir, we have to type our reports on Word, MS Word means.
आपको प्रिफरेबली अब आप बताइए कि वर्ड, फिर एक और व्यक्ति सवाल न पूछे कि 97 में करना है कि 2003 में करना है वर्ड के आप वर्ड में टाइप करिए और किस में टाइप करना चाहते हैं आप कोई बहुत स्पेशल कोई चीज में टाइप करना चाहते हैं
No sir, just confirming.
आप कोई भी इजी सॉफ्टवेयर में टाइप करिए और सबसे आसान सॉफ्टवेयर वर्ड है ना. There are complex softwares available which allow you to draw and do things like that. I have no problems. You want to convert it into a PDF, you can submit it as a PDF. You can submit it as a Word document itself. At its worst case, you can write it by hand and take a picture and submit a PDF. But all of it has to come as a file in Moodle. Okay, and it has to come before next Monday. Okay, because otherwise you will be marked absent. There is no such thing as doing it at the last moment. So all of you, please submit it on Moodle and ensure that you are there on Moodle. Shall we start?
Yes, sir.
Okay.
Yes, sir.
Right. So how do you want to do this? You want to push this off, put this off the whiteboard, or you want to use the whiteboard?
It's fine. We can use that.
You want to use the whiteboard also? But how will you show the pictures? So you will put the camera on, and then you'll have to come to this place
We'll
here.
first share the presentation and then.
You want to? How do you share? Okay. Start.
We can manage.
Just wait for some time. Let this get sorted out because my laptop is being used along with their own. So there's some matching has to be done. So I'm sharing the screen, so you can minimize this. Minimize this here. Don't dismiss. Don't dismiss. That's just recording. This you can also minimize. You want the screen. You can minimize this also. What do you want to do?
I just wanted to stop the sharing.
Stop the sharing.
Yeah,
Then click on this. Click on this.
Yeah.
Yeah, and you can stop sharing. It's here.
ठीक है, अभी stop.
Stop. But they keep on asking whether you are being heard or not from time to time. Can you hear me?
Sir,
You should ask.
Yes, you can ask.
Sir, do we need to make groups for this practical as well?
Everybody has to do an individual thing. The groups are required because the number of equipments are less, and therefore by definition you are able to not able to do the experiments individually. Here you are observing experiments and therefore there are no groups. Each
Okay,
has
sir.
to submit individually. Yeah, there is no such thing as a group. You have to go to the screen and start sharing again.
So can we start?
Yes, sir.
Okay. So last time we shared some slides with you and regarding the basics of this yeah, E. coli culturing and how to use the the laminar hood and all those things. I hope you remember all those things. So any for just before we begin, so are there any queries or questions you want to ask regarding what we have said to you last day? So no questions. So I hope we can proceed with today's session. So okay, let's start with it. So today's lab session is about recombinant protein expression, and I hope your assignments will be based on what we are going to share with you today, and we will also let you know, and we will also communicate with sir that how you can prepare your assignments based on what we are talking or what we are discussing today. Is it all right?
Yes, sir.
Yes, sir.
Okay, fine. So the first thing we want to tell you that, so you are familiar with how many in silico softwares or platforms you are familiar with that assist in making clones and cloning genes, etc., etc. Because basically, first what we do is that before we start any, we can divide the whole work into small projects actually. So your cloning of a single gene can be considered as a project. So unlike from older days, what we do now is that we first make a project in the in silico using computer and softwares. We verify it, run the in silico gels, and see whether what can be the probable fragments and all those things, and then we do the whole thing in silico first, and then we start our session after that, the hand session. So basically, the in silico part actually helps you to narrow down many problems, and at the end of the day, when you go to the lab and on a workbench, you just have to do the pipetting because all the rest has been already planned and done in silico. So the in silico part is very important in today's cloning and all kind of molecular biology work. So you familiar with any softwares or in silico processes which assist in cloning or which helps you to find out primer designing and all those things? Anybody
Yes,
listening?
sir. We had one program in recombinant DNA technology.
No, no. I am asking, are you familiar with any in silico platforms that are used for cloning or that are used to demonstrate how a DNA can be cloned or like that, or how primers are to be prepared? What will be the properties of the primers? So are you familiar with any of softwares or an online platform like this?
So just a gene runner where we can design primers.
Sorry, can you repeat that once again? I didn't get it.
We just learned about a gene runner where we can design primers.
Okay, so you are not familiar with
Sir,
like,
I can name one. Sir, emboss.
yeah. So emboss, what you use? We have like we use the emboss needle for blast, right? Hello.
Yes.
Okay, so there are options like P blast. There are options like nucleotide blast. So these are blast softwares. Okay, so uh, these are used only for like whether to see that how there are many types of work you can do. First of all, that blast is actually used for, it is an actually advanced version. So we are talking about some basic software, basic platforms that need to be handled in order to make your project fast. Okay. So we will go, first tell you about some of these softwares, and please participate, because else it will be very difficult if we keep on telling this, and we will communicate it to sir, and then we will ask you to make a project and you will not understand anything. So please communicate is very essential, at least for us to know whether you are understanding or not, right? So first, you all know that protein. This is the typical this protein purification and protein expression. So protein expression is nothing. It is actually proteins are the terminal downstream product of a gene. Okay, so all genes are converted into RNA, and RNAs are converted into protein. So all RNAs actually are not converted into protein. Some makes regulatory RNAs, some makes different other things. So, but proteins are the terminal product of the genes, for the genes which are responsible for encoding a functional protein. They are translated into a protein. So proteins are of various for the various reasons. Protein are very useful for us. It is actually the machinery of the cell to perform all kind of biochemical works. Basically, all the hard hitting works are done by the protein, and they are involved in almost all, from performing structural features to have metabolism and protection. All are dependent on protein. So the basic first thing about understanding protein is to have the protein on your hand. So how do you study a protein? So you have to have that protein in your hand. So previously what people used to do is to that, like they used to purify the proteins from animal tissues, and the purification process were very inefficient. So you may end up with 100 kg of meat from which you just recover 1 or zero point 0.5 milligram of protein. So that was the rigor that was required for protein purification. So after that, what was discovered, that since the cell is highly a modular thing, the modular thing means that it's like a car. You can deconstruct it into single components, like the engine, the battery, the steering. You can just deconstruct the whole car into single components, and if you put them together again, the whole car will be there. So the cell is also a very modular thing. So you can deconstruct it into single part, and that single part can be transferred into another machine where it can perform the same function. So you can take a gene from any organism, and you can clone it in a different organism, and that organism will also produce the same functional protein. It may sometimes not be functional because there are folding issues and all those things, but the gene will be expressed and it will be the same protein. The amino acid sequence will be the same. So this was the idea which was implemented, and the idea was implemented in order to uh, and to implement this idea, what people learned was cloning, right? So cloning has to be done, and how cloning has to be done, that was a challenge, because it is actually difficult to clone something in a complex organism like a human cell line or something, because there are two copies of chromosome, and each there are two copies of each gene. So where you want to clone it so that the expression comes. So then people narrowed it down to the bacteria which have a single chromosome or a single genome. So anything you clone inside a bacteria will produce a protein of your interest. But it is difficult to clone any gene into the genome because the genome is very large, and you have to figure it out how to get this where to clone and how to integrate that. So in order to solve that problem, people came up with plasmids. So plasmids are mobile genetic elements which can be acquired by the bacteria through horizontal gene transfer. It can also be acquired by vertical gene transfer, but in lab, when we express any protein, so we express it into a plasmid because the plasmid can be transferred horizontally. So then we talked about what is the sources of the plasmid can be different, and each plasmids have different parts as you will learn it theoretically. So we will tell you here how to clone a gene inside a plasmid, because at the end of the day, it is the plasmid which will express your gene. Any questions till now?
Sir, I want to ask some question. Sir, you said here we will teach that how to clone a gene in a plasmid. So we will learn it in silico, or we will learn it biologically.
So that was what I said into the introduction, that nowadays, before that, people has to do a lot of trial and error and experiments in order to clone a gene. Now what we do, things has advanced and we have the in silico platform. So whenever you clone a gene, or whenever you want to express a gene into E. coli or any organism, first you take it as a project. You deconstruct the project into individual components, and the first part of that project will be to understand it in silico. So you design the whole experiment in silico, confirm what are your probable results, you write those down, and then you go to the lab, and then you work on the bench. So you spend 80% of your time designing it in silico and just 20% of time just cloning it and doing it biologically. Is it clear?
Yes, sir.
Is it clear? Okay. So so now, as I told you that these are the parts. So now Manjul will tell you about the about where where will you get your vectors and how to start the cloning aspect, and she will walk you through all those things parts now, and I will also join after this. Now you just over to Manjul.
Hello guys. So this is the picture that we need in at last, protein expression, a perfect gel picture with ladder and our protein expressing. So you know it comes from a functional gene, and so there are two main aspects of the protein visualization on the gel. First is the gene cloning, and second is the expression part. Gene cloning, why? Because we need a gene that is producing our protein. Are you guys listening now?
Yes, ma'am.
Yes, ma'am.
Okay. So gene cloning is one aspect, and then we come to the when we have the cloned gene in our plasmid, we express it in our host so as to produce a protein. Fine. So why do we need to clone a gene? Why do we need to express a protein? Why do we need to express a gene fragment? Is because at times we need to know about the function of a gene. We need to study what is the function of the gene. If we knock it down, if we knock it out, or if we express it in excess. Second can be our strain modification, like there is some bacteria or mammalian cell, we need to modify our strain so that it produces more of our gene of interest. And next can be the protein production. There are certain proteins which are enzymes or antibodies which need to be produced on large scales. So these three can be taken as our main driving forces for the gene cloning. How is a gene cloned? The main ingredients of gene cloning are like vector. There are different types of vector or plasmid that you call. So vectors can be divided on the types based on our purpose. Like cloning vector or expression vector or gene knockout, gene knockdown, or anything like that. Our purpose can be different, but here we are mainly concerned about the expression of a protein. We need to see a protein which is expressing in our host. So we will go for a expression vector. There are cloning vector and expression vectors. Cloning vector is a vector in which there is no promoter. There is a gene which is cloned there, and we need to save our gene. So we clone it in a cloning vector. And the expression vector is where we need to see the actual expression of our protein. There is a particular promoter which is activated at induction, and it gives the expression of the protein. Transcription unit is there, promoter, transcription, stop codon, everything is there, so that it makes a functional gene which is expressing our protein.
When you said that
So
in cloning vector promoter is absent, so what is the significance of cloning vector? Because if we are not expressing the gene,
okay, there can be certain genes which are hard to clone actually. So we need to save the gene. We don't need any expression, and at times there are the leaky expression of some of the these all genes now. So we just need to save them. We do not want to express them in any of the vectors. So that is just for the cloning purpose, that it is cloned here and saved there. Do you get?
yes, ma'am. Yes, ma'am.
There are also vectors which are shuttle vectors which need to express in different types of what you call that organisms. So cloning vectors are only there just to save your gene of interest. It is there and cloned, and you have saved it for future use, or you have just cloned it. It is just for the purpose of cloning. Okay.
Yes, ma'am.
Next is our gene. Okay, next is our gene fragment that we need to express the protein, which is about which we want to study. So that is a gene fragment that we need to define. Next is we have plasmid and we have gene. We have to combine them in a single functional unit. So what do we need for that? Restriction sites. We need to cut and paste gene from one site, and we need to paste it in our vector, expression vector. So what are the basic steps that we need? What are the basic things that we need to remind us is the restriction sites. Do you understand restriction sites?
Yes, ma'am.
Yes, ma'am.
Okay, so the restriction sites should always be absent in our gene of interest. You can see a picture here. This is the gene fragment, and NDE1 and BAMH1. These are the two sites which are present here. So if we add enzyme ND1 and BAMH1, this will cut our gene in between somewhere. And if we clone this fragment, first we won't be able to clone this in our vector, which is cutting with same ND1 and BAMH1. And if we clone, we will clone the wrong fragments. The whole gene would not be paste there. We won't be able to paste the whole gene. It would be a fragment of the gene, and we won't be able to get the expression of our required protein. So the restriction sites must be absent in our gene of interest, but they have to be present at the end of the gene of our interest, so that we can cut them and we can paste them in our vector. So restriction sites have to be absent in our gene of interest. They have to be present at the end of the gene of our interest and have to be present in our vector. Next is the expression host. We need to choose which expression host we are going to study our protein of interest. Whether it is a bacteria, it is a mammalian cell line, it is an insect cell line, fungus, or Pichia, or anything. According to our expression host, our vector also differs. There are vectors which are only expressing in bacteria, which are only expressing in mammalian. So we need to choose our vector or the plasmid for our purpose, whether it has to be cloning or expression, and whether which host we are going to express it. This is the simplest gene cloning that we can think of. This is a plasmid which is cut by two restriction sites. At the ends, it has been linearized, and our same gene of interest, this has also been linearized with the same restriction enzymes, and then ligation with the help of ligase enzyme. These have been put together in one single recombinant DNA. Now this DNA we have to transform in our host, whether it is a bacteria or mammalian cell line, and we have to check for the expression. Any questions?
No, it's clear.
You can also get the vectors. AdGene is a vector database where you can know about the different vectors, about their properties, where from it you can get it, what is the name, the whole sequence, what type of vector it is. It is expressing in bacteria or in mammalian cell, or what the promoter type it has, what resistance is there, ampicillin resistance, kanamycin resistance, or any antibiotic resistance, whatever is present there. Everything you would get to know about the plasmid from the AdGene. It is a vector repository. Next is we come to the protein expression. We need to show them this inoculation and everything, how to.
So you can turn on the camera.
Stop presenting.
Then now you can show the camera is on.
Okay. So for the protein expression thing, now we have our host ready. We have transformed our cells and we get a plate like this, single cell, making a single colony. Are you guys? Can you see the plate?
Ma'am, can you just expand the screen?
Okay. Now, is it okay?
Ma'am, I guess you can stop presenting. It'll be clear.
Okay.
इतना ही होएगा.
Now, can you guys see the camera? My camera is on. Can you see the plate?
Yes,
Yes.
ma'am.
Okay. After transformation into the host cell, this is the type of plate we get for our clones. These are the single clones which have our recombinant DNA, the plasmid containing our gene of interest. Okay. These single colonies are picked and we make single clones like this. Okay. From this plate, we inoculate into a 5 ml culture tube a single colony for our protein of expression. These clones are the ones which have our recombinant DNA having the plasmid and our gene of interest. We have confirmed the clone after taking out the plasmid from a plasmid DNA isolation kit, and we have checked with the restriction digestion, whether the vector size is proper or not, whether our insert size is proper or not, or if it is getting any other DNA contamination or not. So these are the confirmed clones which have been confirmed after restriction digestion, and they have our recombinant DNA. So a single colony is picked from this, either this one, or this. A single colony is picked from this one and we inoculate in 5 ml culture. We keep it overnight shaking. This is our primary culture, which is grown overnight at optimum temperature. It can be 37 degree, 25 degree centigrade or 30 degree centigrade according to the nature of our protein. From this primary inoculation, we inoculate in 2 flasks. 2 flasks. One we will take as a control and the second will be induced. There are promoters. Okay, we are inoculating two flasks. After these cultures are in early log phase, we induce one of our culture. This is our induced flask. Why induce? Why induce? Why do we need an induction? We need an induction because there are proteins we need to study about if there is any leaky expression or not. For that, we need to there are different promoters. I'm sorry. There are different promoters like T7, ARA promoter, PBAD. All these are the promoters, which are the control of which are under the control of some of the activators. They are inactive until they are made active by the addition of some of the induction. So when the culture is in its early log phase, we induce one of our flowers, and after that we uh, collect sample from time to time. Is that clear?
Yes, ma'am.
Then what is the purpose of control? There are two, control and induction. So what is control?
Okay, there is a control we need to, because uh, the protein of interest, the protein of interest that we are going to study about, if suppose it has a 60 kDa size, so in cell there are different proteins which may be of 60 kDa. So do not expect a single band of your protein of interest in this thing in your cell culture. So when we compare uninduced versus induced, we get to know if there is a band intensity more than as present in the uninduced one. We get to know कि okay, our protein of interest is getting expressed. After that, we need to confirm it with different methods. But this is the preliminary step that we need to do. That's why we compare uninduced and induced culture. Got it?
Yes, sir. Yes, sir.
Okay. Arun. Guys, wait, I'm presenting my screen.
ठीक. So I hope you got how to express the proteins and all those things as shared by Manjul. And this was the last slide because these were the kind of promoters like we use in E. coli. And now I am going to show you something on the in silico part. And these Professor KJM may actually put these things as your probable assignment task. So I'm going to share that thing. So please give attention. Are you guys there?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So the important part is that now we are going to the in silico part of it. 200 microliter add करके 3 बार wash. So we are going to the add gene. So so I will tell you first. First, I will explain you with a question how the questions can be framed. Okay, for your assignment. So you can be asked like you can be asked like the add gene is a repository of vectors as Manjul has told you. So there are various kind of vectors you will get at add gene. So one question I may ask or Professor Mukherjee may ask is that each of you pick up a vector, you open the sequence of the vector and from that sequence, write the sequence of like the promoter which is driving your recombinant protein expression for that vector. Is it clear? The question is clear?
Yes, sir.
Okay. So in order to understand that you are not copying, we may also ask like,
Sir, can you please
you have
repeat
to
one?
write. Okay. So the question is like that. There are many vectors in AdGene. So you may be asked that take up one vector, you open the sequence of that vector, analyze the sequence, and write down the promoter sequence that is driving the recombinant protein which the protein which has been cloned in that vector. So you may be asked write the sequence of the promoter for that vector, and in order to confirm, because most of the vectors have the same promoters which are driving your gene of interest, you may be asked to clone write down the sequence of the promoter with 10 base pairs ahead upstream of that promoter sequence. So in order to answer how this in order to solve these problems, I am going to show you how this has to be done. Okay, please pay attention.
Discard and then
हाँ, discard and add. आपने इसमें वो किया था क्या? So now I am opening the sequence of a PET22 vectors. Now you can see there are many entries for the PET22 vector. Like, okay, so I can like pick up any of this. So this is plasmid. This is the catalog number. So you don't need it because it is useful for ordering. So this is the PET22 vector. So we call it the backbone. So this is the information about the backbone, and this is the information about the gene which has been cloned in the vector, right? So now you can see the purpose: expression of a single cysteine variant of a mature phosphate binding protein as a scaffold for phosphate biosensor. So this guy has used this PET22 vector as a backbone for cloning this particular gene and for expressing that gene. Now you have the other information. What is that? This is the name of the insert that I have already described. Now the expression is bacterial. So bacterial means it's mostly E. coli, so it is not separately mentioned. If it is other, it will be mentioned. The name of the promoter which is driving this gene which is cloned here is the T7. And now you have the data availability that this is for academic institution and non-profits only. You can order that and you can obtain the plasmid and do your work, but it cannot be directly used by commercial organization. So now if I click this part, now you see that we come up to this page. So now what we have is that this is the whole map of this thing, right? And now it is showing different components. Mostly they are restriction enzymes. This is the functional units. This is F1 origin. This is the promoter and all these things. You will learn it theoretically. I'm not going into the details of that. Now what we can do is that we can go to this part, view all sequences, right? So now they have provided you the sequence. So you can go to each and every individual sequences. There are multiple sequences done with it. And these are the information of the primers which were used for sequencing this particular vector. So this one, these are fragmented sequence. And you have here, this is the whole sequence, right? So this information provides you what is the total size of the vector, means how many base pairs are there. So there are 6339 base pairs in this plasmid whole construct. Now if you come here and click at the analyze sequence, So now a different you land up in a different page and where you get the same picture map here, and now there are different data tabs over here which you can explore. So I am going to the sequence part first. Now you can see that the things which were there in the map are also highlighted here. This is the part of the origin. This is the AMP promoter driving the ampicillin resistant marker. And so as you go down, you will see different things which are the part of the vector. Now we are particularly interested in understanding the part which is driving our gene of interest in this vector, right? So as you come here, you see that this is the LAC-I promoter. Okay. And this is the T7 promoter. So the T7 promoter, after that there is a LAC operator, and this is the ribosomal binding site. And now here you can see this is the start of your gene, right? So all bacterial expression starts, mostly 99% cases are with ATG. Some starts with GTG. So wherever you see an ATG after the RBS, so that is the gene start site. So we may ask you like, we may ask you the question like take a PET22B vector and from the add gene, any gene you cloned you want, and then write down the sequence of the promoter, right? So what you have to find out is that these all sequences are given in 5-3- direction. So we are only considering the upper strand. We are not considering the lower strand. Whenever you will be asked questions about the sequences, you have to always look at the 5-3- direction. So that means the upper strand. So when you will be asked like what is the write down the sequence of the T7 promoter, so you will have to consider the upper strand, that is 5-3-, and then let me comment here. Yes, and then if you click on this part, you see the promoter sequence has been highlighted. So when somebody asks you what is the T7 promoter sequence in this particular vector, so you have to write down these sequences TAA, TAC, GAC, TCA. And when somebody asks you that, you have to write down the promoter sequence with 10 base pairs upstream, so that you have to count from here up to 10, and you have to write down the whole sequences up to here. Is it clear?
Yes, sir.
Yes, sir.
Right. So whenever you clone something or whenever you talk about a DNA strand or something, you have to always look at the 5-3- direction, right? And whenever we talk about sequences, whenever we talk about you have to write a sequence, you have to always look at the 5-3- direction. So so there are two 5-, and we will always consider the upper sequence. This is, I mean the upper strand, not the lower strand. Got it?
Yes, sir.
Okay, so now to do it intelligently, what you can do is that.
So I had one question.
Yeah,
So in the
please.
T7 promoter, we can see there is one guanine which is a part of both the T7 and the LAC operator, I guess. So like the last guanine here.
Yeah.
Yeah,
Okay.
in the purple arrow.
Yeah. So you are talking about this
So
G.
uh, yes, sir.
Yeah.
Primer है उसको.
Yes, sir. So
Ah.
so how do we consider this as a part of the promoter as well?
So there is a click. There is a actually thing to it. So whenever you see that here it is written promoter, right? So whenever you click this, so this part is highlighted, right? But this is not the promoter sequence. This is a primer sequence which has been given here. Because if I want to put a primer over here, I can. So these all things are constructed using a software called the SnapGene. So in SnapGene, you can use different functions. So if I want to say like like somebody wants to tell you that please, I have to clone this part with the T7 vector. So we have to amplify the gene. So how do you amplify the gene? We use a primer to amplify the gene. And how do we construct a primer? We can highlight the primer over here using that SnapGene software. So what they have done is that they have done a sequencing of the cloned gene using a primer. and they have highlighted the primer over here. This is the notation of a primer. This is not a notation of any functional component of the vector, right? Whenever you see notation like this with a half-headed arrow, this is the primer. Clear?
Fine, sir. Yeah.
Okay.
Thank you.
So whenever somebody wants to tell you about a T7 promoter, you have to click this part, not this part. So this is the primer. And another thing to add, that it is unlike the mathematics where you have to be exactly precise about the figures. Here, if you like miss 1 or 2 nucleotides, it doesn't matter, because the person who will be looking at your assignments, he knows that these are common mistakes that can be ignored. But grievous mistakes are something which has been, like, if you like write it up to here, then it doesn't, but it clearly shows that you haven't understood it. But if you take 1 or 2 G or G, if you take this G or an A letter, it doesn't matter, because the person understands very much that is okay. That means that he has actually written 2 nucleotides extra. So that doesn't matter. If you if I tell write 10 base pair from 10 base pair upstream to the last one, and if you take it from like T, it's okay perfectly. There is no problem about it. But until and unless you are sure and you are understanding what is the region of interest rather than the exact nucleotides, you have to be you are okay with it. Okay, so what matters here is the region of interest, whether you are getting the getting an understanding of the region of interest here. Am I clear?
Yes.
All clear.
Okay, so when somebody wants to ask you, write down the RBS, write down the sequence of the RBS, you click here and you highlight here. Now, if you say start with AAT and you end up here like after T, it's absolutely fine, because you are actually highlighting the region of interest, that is the RBS. The RBS is the ribosomal binding site. Okay, so you can click over here and you can highlight the RBS, and now you can exactly come here and you can select it, and you can control C, you can put control C, and now your RBS has been copied, and you can now paste it anywhere you like it, and this will like your RBS part will be copied. Where is this presentation going? So for demonstration, I am showing you that I have copied the RBS. Now you see. You can see the RBS part now. I have pasted on the presentation.
Yes, sir.
Yes, sir.
So that's how you take up individual components using that particular thing, and you do it. So sir can ask an assignment like this, that you have to take any vector. Each of you take up one vector, write the name of the vector, list the functional part of the vector, and then he may ask that write down the sequence of the T7 promoter or the RBS for the vector, or write down the sequence of the gene of interest which has been cloned here, right? So whenever for the gene of interest, you have to look at this. There's an RBS, and after that, the first ATG. The first ATG, if you look at so this is methionine. Okay, so any translation starts first with an methionine. So you have to look at the methionine, and if you click it here, so you are seeing that this methionine is coded by the ATG, and it is highlighting the ATG. So that is your translation start site. Okay, so your polypeptide or the protein of interest which will be expressed will have will put its first methionine from this ATG part. Okay, and so you can also, like, if somebody wants you to tell that you tell me the sequence of the gene. It's a bigger one, I understand. I hope he will not ask, but still I was showing you. So you can actually copy this whole gene from here. Mm, mm. No, I think it's not possible because it's not apparently it's not allowing to copy us. So I hope I will tell him that it's a problem and we cannot do it further. So you can do only for those things that the properties which are actually highlighted. Like you have the 6 his, so this is the his tag sequence. Okay, this is the tag for purification. So this is the T7 terminator. So you can put a click on this and highlight these things, and you can then after it is being highlighted, you can press Control C directly, and you can directly go to the presentation part and or anywhere you like, and you can like if I am inserting. Is it a problem with the internet? Why it is not copying? I don't know how this laptop is functioning. There is some problem with this. So okay, so I have given you the introduction. Okay, there is some problem actually we are facing. I don't know why it is not copying. It usually is good to copy, and I don't know if it is Team is interfering. And we will also show you the mother software using which these gene files have been prepared. Okay, so that will be for the next class. But right now I hope that you understood that how to find out the parts which are actually required. So any questions regarding this, any clarifications? Any questions? Any questions regarding this?
No, sir, it's clear.
No, sir.
Sorry?
You guys are clear about your assignment now?
You are all clear about the assignment, na? What I told you, how this can be, how the questions can be projected.
Yes, sir. It's clear.
Yes, sir.
Okay, so in that case, matlab, nobody should tell like we didn't understand. If anybody have any question, you can ask me now. Okay, fine.
So when will this assignment be given? Like, and is it
Sir has
separate
already
from the
given you.
lab report?
No, this will be like I think these he has asked you to make a report of the classes, right? So you have to write a report on the classes which includes our PVX class and these class, and we will communicate to sir today that we have taught you this, and then you expect to this assignment to be your next assignment after this one. Any
So,
questions?
like, so for the next class, what are we supposed to submit? Like, for the next week's class, before the next week's class, what exactly are we supposed to submit?
So for the next week class, what we have taught you last time is that we gave you some basics, right? About the laminar hood and all those things. You can write a like small short paragraph about the summary of the whole thing that we told you in the last class. For this class, you may you have to go you have to first write a small summary within 200 words about what we said, what we summarized, and then you may pick up a plasmid from the ad gene. You write down the name of the plasmid. You write down the you write down the name of that plasmid, and then you say mention what is the uh, so what is the entry number for the plasmid. You mention that, and you may summarize that what are the genes that what is the gene that is cloned, what is its origin of replication, what is the antibiotic resistant marker. So we want to keep it simple for
Sir, do
this
you
one because go ahead please.
sir, do you have a sample report or something like that for this? Like, or maybe the assignment written, like the problem statement and.
No, I don't have that. That sir might be having. If there is available any, but I hope it is pretty clear. Pretty clear. You have to write. You have to take up any plasmid. Like you note this PET22B. You search it on the AdGene. You write the at least the plasmid. This is the catalog number for the plasmid. You write the catalog number. You write in basically which organism is the host. You write down the promoter. You write down what is the gene that has been cloned, right? You can write about it. You can Google the name of the gene and find some information about it, and you write the summary what you understood by what that gene particularly does. You can make a very good presentation with that.
Do we need to supply the sequence information as well?
I think it will be too early. We are not asking to submit any sequence information right now because I think we will take another class based on the original mother software through which these sequences are made, and then we will be able to clearly show you how to copy, how to run a blast and all those things, and we can put it for the next assignment. Expression of protein. Is it clear?
Yes, sir. So basically, the lab report is the assignment, right?
Yeah.
Fine, sir. And like previous weeks and this week's, we have to submit one whole doc, like by next Monday.
Yeah, exactly.
Fine, sir.
Okay, so we are keeping it like simple so that I hope many of, since you are IIT guys, many of you must be knowing many things, but some people may not be knowing. So let us keep it equal so that we can move step by step and we can learn. We can better learn rather than try to understand what we know and what we don't know, right?
Yes, sir. So one last thing, where are we supposed to submit these? Because sir said that we have to upload them on Moodle, but since we are not enrolled, like the third year students aren't enrolled in the 433P course, so we don't have a Moodle channel for that.
So you are from first year or second year, third year?
Third year.
It's the third year.
So third year people don't have a channel. That's a problem. That's the problem.
So we don't have the 433P channel on Moodle or on Teams.
Okay, well, I will write it down and I will communicate with him. 433 channel in Doodle.
Same with final year students.
Okay. Final year students. Okay. Okay, I will tell him about this problem. So I hope you got now what we are asking to you in order to how to prepare your presentation. Your assignment. Right. Yes. So any questions regarding this? Because you guys are not asking questions. I think don't you are you not finding it interesting? Or you can also request something of your interest what you want us to also tell you. That can be also very nice.
So you said that.
So I guess once we.
Just a second. So I was asking that you asked us to mention about the origin of replication. So what do we write in it? As in the sequence of the origin, or what do we have to mention in there?
So, okay. So now you open this file. So you see that there is one word written F1 ORI.
Yes, sir. And there's another ORI there.
Okay, so if there are two ORIs, so you mentioned that there are two ORIs and one is F1 ORI.
Okay. Got it, sir.
Okay, so I am not describing the two presence of two ORIs because I hope these are things which are theoretically covered in your classes. And if you don't know, you can ask me. I will again. I will also explain this F1 and two ORIs. Okay.
Sir,
So any.
I had a question, sir.
Yeah, please.
Sir, in the inoculation plate that ma'am showed previously, are all the colonies having the same vector and the same insert? Is it
Yes.
is it the same all
Yes.
throughout? So
Yes.
then why is
Because
it making so many?
okay. So the thing is that what we say, what we mean by the word clone. The word
Yes,
clone itself means an exact copy of each other, right?
yes, sir.
So what happens is that see, I you have an understanding what is the OD of a culture? What does an OD of a culture means?
Sir, when we look at it through the spectrophotometer, we want to know the density, cell density.
So
So,
that's how we measure it. But what is it? What is OD?
so absorbance.
No. Any of you have any idea what is an
Optical
OD of a culture?
density.
That's
What does it reflect? What does it means?
You are telling the full
Only
form of
the
an
cell.
abbreviation.
Yeah, this. I mean, it probably shows the number of cells that are in the sample, right?
Yeah. So why you are using the word probably? Don't use any word like probably or may. You have to be absolutely sure. Yes, it means the number of cells in your culture, right? So
Yes, sir.
one OD means 10 to the power 8 cells per ml of culture, right? So now when you are sorry again. Again, please, if somebody have a question. Okay, fine. So one OD of culture means 10 to the power 8 number of cells, okay, per ml. So what you are doing that you are adding a plasmid. Okay, so what say you are adding 1 nanogram of plasmid. Fine. So you are understand now you have to calculate like these are when you will move along and if you are interested to study molecular biology and all those things, you have to calculate how many number of plasmid molecules are there in 1 nanogram. So say we have a plasmid plasmid which has a molecular weight something say something like 5000 kilodalton. Okay, so what does it mean that uh, in 5000 kilodalton, 1 mole of that plasmid contains 10 to the power 23 number of plasmid molecules. So 1 nanogram of plasmid molecule will corresponding to certain amount of number of plasmids. So say there are 10 to the power 5 plasmid molecules or 10 to the power 8 plasmid molecules in 1 nanogram of the plasmid you have added to the culture. So now when you are providing the heat shock or when you are giving the electroporation, so there is a probability that how many plasmids will make it up into the cell, right? So if there are 10 to the power 5 plasmid molecules in the culture and if you are providing a heat shock, so you can expect roughly 1%. That is 10 to the, if it is 10 to the power 5 number of molecules, 10 to the power 3 number of molecules will be transformed into the cells. So after this, the plasmid has gone inside the cell, all cells may not survive. So again, there are 2 to 3% cells which has been transformed with the plasmid will actually survive. And then if you have if you have like 2%, so how many of these are of the, so if you have 10 to the power 5, so 1% means 10 to the power 3, and then 2% of that, how much does it, like I think it's 20. So now you will get 20, there are 20 cells on the plate which will now divide individually and make up a colony. So each colony represents an individual cells who have divided multiple times in order to make a colony, right? So roughly 20 colonies means 20 cells have survived the whole process you have done. and so the plate contains like 20 colonies. So if your competent cells are very good, so you may end up with 1,000,000 colony on a plate because your cells were very good and most of the cells got transformed. If your competent cells are not good, you will end up with very few colonies on the plate. So that's why when you see the plate ma'am have shown, you see that there are only so many colonies, and obviously you're interested that I just need only one, but you cannot have a single colony, right? That's a process.
Yes, sir.
Okay.
And also we need to screen for more than one clone. We need to sequence more than one clone so as to check whether our protein sequence is correct or not, because there might be insertion of bases also.
Yeah.
So
yes.
Is that the reason why you divided the plate into different cells?
Yes.
Typically for every clone we streak 10 colonies, individual colonies, because you see it is a bacteria. So bacteria has a very fast rate of evolution. Okay, so many of your many of the clones will have actually a mutated gene. Okay, you can end up with a mutated gene. So what we do is that we maintain 5 to 10 colonies at least, so that so that the clone we are selecting is actually what we wanted, not a mutant one. So if one actually turns out to be a mutant, we will discard it and we'll take the rest, because it is actually very difficult to have all the 5 actually mutants. It's not possible. So there are the probability says that if you have 10, you will have at least you will have actually one cell that has been mutated. We will discard it and we will have the rest of the 9 fine. So that's the logic.
Understood, sir. Okay.
Okay. Okay. So any more questions?
Sir, I had a question.
Sir,
no problem.
Sir, I can't understand that you said if a large plasmid go, then all the cells will not survive. Only some of the cells will survive. Sir, means why it is so?
Because survivability in nature is a statistical phenomena at all. All human babies don't survive. All dog puppies don't survive. So similarly, all bacterial cells which have got transformed will not survive.
Okay, sir. Okay, sir.
Sir, I had a question that you said that we are getting a single colony from a single cell. So why is there is not a possibility that there would be some plasmid-free cells in the colony? Like why the
100%.
plasmid-free cell colonies are not emerging in the
colony? So that is possible. Your colony is dividing again and again, and some cells may lose up the plasmid. That's absolutely good. But you are taking that colony and sticking it again on an antibiotic plate. So obviously the guys who are not having a plasmid will die.
Okay, sir.
Right. So that's why researchers do multiple streaking in order to ensure that only the cells which are having a functional plasmid actually gets selected for the next step. Clear? Okay, fine. Then we are ending up this class here. I hope you guys are satisfied with whatever small things we were able to offer you, right?
Yes. So also please, if possible, convey the message to sir that like the third year students aren't added to the Moodle channel for 433P, and we aren't added to the Teams channel either, so we can't access the recording of the last session which we couldn't attend.
Fine. I will communicate.
So same for final year students.
Final years. Okay, and you can also if you like, you can also give a feedback to sir if you are liking our class, and if you like also it's good. If you don't like also, please tell properly, because that is also important, right?
Sure, sir. Thank you, sir.
Okay. Bye. See you all.
Thank you.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir.